It's my pleasure to introduce Dr. Hope Jahren, who is joining us from the University of Hawaii, where she's been since 2008. Prior to that, she was a professor at Johns Hopkins University, where she started her career as a faculty member in the field of geobiology. If you joined us last night, she gave a really interesting talk about global change and our food supply for our hungry planet. And today she's going to talk to us about what the isotopic composition of plant tissues can tell us. So thank you.
Okay, thank you very much for helping me. I'm Hope Jahren. I'm visiting you from the University of Hawaii. I want to say a special thanks to Keelan, to Ashley, and to the other students that work hard to put this seminar schedule together. They were very, very good hosts to me, and you should be proud of them for taking the initiative to make this program and to invite people and to treat us so terribly well when we do visit. It's a great thing for Utah State, and you are lucky to have them. Yes, okay. A little bit about me. I am a professor at the University of Hawaii. Before that, for 10 years, I was a professor at Johns Hopkins University. Before that, I was a professor at Georgia Tech. We have been working on a variety of problems, but the story I'm going to tell today is about our journey through a problem for the last 20 years. And we think we've arrived at something that's pretty significant. But who are my students and postdocs in the audience? Right, okay. So when I talk to people about how to give a talk, because a lot of your career comes down to a 1-hour talk. You know, you may be at an interview or you may be at a conference or something, and you're often dealing with people who haven't had time or inclination to read your papers. And so an awful lot can hinge on a 1-hour talk. So how to make a 1-hour talk good? There's a lot of different philosophies about that, but my own personal philosophy is that you should tell a story, because seminar and talks are really the oral tradition of science. And so today I'm going to tell you the story of the last 20 years of our research. You're going to hear about things that we tried on a whim. You're going to hear about mistakes that we made. You're going to hear about times when we pulled back and did things over. And you're going to hear about some of the successes that we've had. So let me show you 1-hour highlights of the last 20 years. It hasn't been just me along the way, and I want to point out there's been an army of people. I think we count up 40 undergrads and 12 grad students and 8 postdocs, something like that. But I want to point out just a few people as being very special for this work. Brian Schubert was my postdoc. He did a lot of the mathematical modeling that you see today. And he's a really special person. You should have him here for a talk. He's now a professor at the University of Louisiana, and he's running a big lab like mine. Bill Hagopian is my lab manager. He runs my lab. He was an undergraduate in the same lab where I was a graduate student. So we've been working together for about 25 years. During the early parts of this work, we're at a place now where we have a lot of support to do this work, but during the early phases, when I was a starting out professor, we lost funding many times. There were times when I could not pay him. He moved into, for 18 months, he moved into his car and lived in the parking lot and said to me, Hope, we're doing something really important. I believe in this stuff. We've got to keep it going. It's going to be good stuff. And so there's no real way to thank somebody like that, but to go around the country and say, you know, this is a great guy. But I also want to share that because science is really more than the subjects we study. It's about people. And I've been really glad to have incredibly dedicated and loyal people working with me on these problems. Ben Check, who was here last night, just for the heck of it? Yeah, so the potato work that you heard about last night was done by Ben Check. I'm not going to talk about potatoes today, but I like to highlight him because he was such a role model within our lab, the most craziest, hard-working guy you will ever meet, who really gave himself completely to science and did a Herculean amount of work. I always go on these things. People run up to me later and say, I want to be your grad student. I want to work on this. I say, no, you don't. You know, before you do, talk to this guy who slept next to a greenhouse, next to a greenhouse for 12 weeks and worked probably about 18-hour days the whole time and came up with something truly, truly groundbreaking, you know. So good science really requires that kind of price, is one of the things I want to say to you today. All right, on to a little bit lighter topic. So the people who were here last night, you're disqualified. Does anybody know where this is? Downtown Logan? No. New York Central Park, awesome. Did you grow up there? No. But this is the Upper West Side looking down towards Midtown. The only thing that you're supposed to get out of this picture is that a world with plants is very different than a world without plants. This is not a particularly dense ecosystem either. This is a fairly obliterated ecosystem in that the density of trees here is actually very low. But one thing I like to start my talks about is that I study plants, I think about plants. Plants are kind of my world, and as researchers, you know how that is. You get very into a subject, but one thing I like to share with people is what the things you learn from being so immersed in something like that. What I notice from people is that we walk amongst plants, but we don't really see them, and we don't really think about them. Again, this is a little bit of a repeat from last night, so I apologize. Most of this talk is going to be all new stuff to you. But I do want to show this slide again. This is basically a bar graph that shows ecosystem type, and it shows the ratio of biomass within a variety of ecosystems, the amount that's plant biomass, and the amount that's animal biomass. So one thing I want to show to you is that we're vastly outnumbered and outmassed in terms of biomass by plants. Most of all, the highest values ever are in the evergreen boreal forests, where this mass ratio approaches numbers like 4,000. But even in places like deserts, places like grasslands are funny because the plants don't ever get really big or make wood. It's kind of an interesting environment that evolved fairly recently, actually. But even in places like that where plants are pretty constrained, those ratios are in the hundreds up to 1,000. So that's one thing that you might not have considered, is that arguably the dominant, successful, and flourishing life form on the surface of planet Earth are plants. Not animals, not microbes, but plants. Or fungi. That's an opinion statement. And it always has been this way. So here's geologic time. This is basically the entire Phanerozoic. So this is 500 million years up to, this is today at noon. Okay, so this is the number of coal-forming sites on the planet. This is from Schubert compilations. You might know his website that shows the continents moving around. These are coals, not in terms of coals that are economically viable, but in terms of coals that represent geologic layers that have accumulated land plant material. If we look around just at, and these are just well-mapped places, if we looked at the number of sites around the world from each of these time periods that contain coals, you see that there are easily many hundred of sites where we can easily retrieve plant fossils from the geologic record. This has been true ever since there have been plant ecosystems on land. So plants colonized the continents, which is a really interesting story in itself. For tens of millions of years, the ocean was florid with living organisms, just packed with life, and land was relatively sterile. Then soon after land was colonized, maybe 450 million years ago, it was only a matter of 10 million years, 5 million years before there were full-fledged multi-tiered ecosystems, bushes, trees, all of the things that we think of as filling up land today. And so there's an awful lot of land plant that has accumulated within the fossil record. Plants in general are interesting. The history of plants in general is interesting. I'm going to talk mostly about the chemistry of those fossils, but there's a few interesting things to think about in terms of how plants are different than animals. The plant fossil record is fascinating because it doesn't contain any dinosaurs. There's no big freaky plant thing that was alive then that we don't have now. There's no trilobite in the plant world. So plants started out very soon. We had mosses. Today we still have mosses. Back then we had ferns. Today we still have ferns. Conifers, ginkgos, horsetails, all kinds of things you can imagine. So land is basically continuing to fill up, to become more and more productive with more and more diverse plant forms. So the history of how plants have thrived on planet Earth is very different than the way animals have. And what does this have to do with? This has to do with the fact that a plant is very different than an animal. And we'll talk a little bit about that later. So here's an example of a plant fossil that I've worked on. I've retrieved plant fossils from places as diverse as these are from Colombia. outside of Bogota. We were working there in the middle 90s, which is actually not a safe place to work in that period of time. So we made a lot of sacrifices to get these rocks. And again, that's part of the game, right? We've also pulled fossils out of much different places, like Ireland. Those were Ordovician. I'll let you click that off. Out of Cretaceous stuff from Maryland, out of the KT boundary, so Cretaceous tertiary stuff in Montana, just to name a few of the sites where we've worked over the years. I'm not going to talk about all those fossils because I can't cram 20 years into 1 hour, but let me tell you about the Columbia stuff. As you can see, this dark matter is basically the accumulation of plant tissues. This was as I was finishing up my PhD, I was simply helping a new professor over in biology, this was at UC Berkeley, unpack this stuff because she needed help. I was unpacking it. I had been working on carbonates, which were all the rage in terms of stable isotopes back in the early 90s. And I was unpacking this stuff and I said, gosh, I bet there's enough carbon in there that we could actually do a stable isotope measurement. And that's how it all started. So we did. We looked at the stable isotope composition of rocks that were of that age, right around the Aptian, which is about 115 million years ago. And this is the carbon isotope composition. It's not super important right now that you know what that value means. But basically, you can think of this as a number line, and my data is this black stuff. And what we saw was that things were pretty stable through the early part of the Cretaceous, and then we saw things take this dive in the Aptian. And I must have analyzed and reanalyzed that point 30 or 40 times, right? And this was in the days before continuous flows. There were no EAs. You did these things one by one using physical chemistry on the vacuum line, right? And then people still wouldn't believe us, and so we pulled out cuticle and we pulled out vitrinite, and again, this was back in the day when you had to accumulate milligrams of this stuff. We isolated it under the microscope and did this. And I want you to remember that I talked about how those fossils came back in about 1996. It was about 2001 before we were able to publish this. So this was a big, big fight getting this through the literature. Basically because before we did this, nobody had worked on carbon isotopes in plant fossils. There was a group at Oxford, Hesselboe and others, that had looked at wood entrained within marine sediments, but we were actually the first people to look at terrestrial fossils. So that's another lesson for young people, is that sometimes it takes a long time and a lot of fairly punishing perseverance to get a good idea started. One interesting thing about this is that it was somehow tied to the isotopic patterns that people had seen in marine rocks, right? In strata that recorded inputs at the bottom of the ocean or in sort of more nearshore environments. So these were actually measurements on carbon, so carbon in carbonate instead of carbon in that organic stuff I just showed you. And this was a measurement on the organic carbon within marine substrates. And we thought that was very exciting, is that we saw whatever carbon disruption was happening, whatever disruption in the carbon cycle was happening, it was not only influencing the carbon cycle in the ocean, we thought we could make a pretty good argument that it was also influencing things on land. One thing that we talked about a lot was why this swing was so much greater on land. And we were very sure in terms of the preciseness of these measurements that that was a real shift, that we were seeing almost twice as much of a smack to the carbon cycle on land as we were seeing in the ocean. And that was sort of the titillating part of how this all got going, is what is the difference between looking on land as looking in the ocean? Because the IODP was active, people were doing marine cores like crazy, JOIDES was out there sailing around, taking new cores all the time. People hadn't isotopically accessed terrestrial substrates. It's interesting to think about that now. Any number of you are doing isotopes on terrestrial substrates that I talked to over the past 2 days. But back in those days, it was a stupid idea that you just didn't do, and it wouldn't lead to anything. Trust me, if I had a nickel for every time I heard that. Okay, so what did it mean? That was our next guess. So then we had to think about how plants really work. So there are some fundamental differences, and I always share this because I think it's fun to think about, and I think people think it's fun to hear about it. So every organism on life, so being alive on planet Earth is kind of a problem to solve. We think of it that way. And in order to successfully persist on planet Earth, every organism out there needs to do, over the long run, 5 things successfully. It needs to grow. It needs to maintain that growth. It needs to store against hard times. It needs to defend itself against catastrophe and pathogens. And ultimately, it needs to reproduce successfully, right? So as animals, and I won't go through the rest of the kingdom, but as animals, we share a lot of strategies toward doing that, right? We have few offspring, but we invest in them a lot, and they have a fairly high success rate. We run around accessing resources. We chase down our food, and we don't have very good climate tolerances. We have to go inside if it gets really cold outside, et cetera. So if we think about how plants are different, we come to some really interesting things. So your average oak tree might put out 10,000, even some years 100,000 perfectly fertile embryos. But less than 1% of those will germinate. Less than 1% of those will even grow for a year. So the statistics associated with reproduction are vastly different with plants than they are with animals. Another way that strategy is different. Plants can't chase down their resources. They have to stay put and take what's given to them, or somehow try to influence the world around them into yielding more, right? Likewise, if things happen that they don't like, if it gets cold, dry, etc., they can't run away. They have to stand there and take it. They have to store against hard times. They have to defend themselves against predators that they can't run away from. And so the strategy for succeeding as a plant is a very different proposition than the strategy for succeeding as an animal, right? Nonetheless, I would argue that they are the most spectacularly successful organism on the surface of the land. So, for example, they live longer, right? They live for 200 years commonly. That's not a freakish thing. You can walk down any street and easily encounter common examples that have lived for 50, 100, 200, 300 years, right? They also grow large. It's not uncommon to encounter a 1-ton tree or a 2-ton tree. So this is commonplace. They live longer than us, they get bigger than us, and they never come in out of the cold. This is really amazing. I only study land plants, and I think that's significant because they're eukaryotes, right? And in the ocean, we have mostly photosynthesizers that are prokaryotes. All that means is that you and I and a plant on land are much more closely related than a plant on land is to a plant in the ocean, to a phytoplankton in the ocean. So I study land plants. I don't know a lot about ocean plants. Because they are very different organisms with very different strategies, right? For example, the average maybe land plant lives for 100 years and weighs a ton. The average ocean plant lives for about 20 days and is 1 cell big, right? So these are very different systems. This is why I work on, again, terrestrial substrates. Okay, enough about me. Let's talk about how plants work. So our job then was to say, listen, number 1, we can measure the carbon in these terrestrial substrates. Number 2, this took years, we can do it right. Number 3, this took years, you get something interesting when you do it. Number 4, what does it mean when you do it? And that took some time. Okay, so we had to go back to how do plants work. So every land plant works approximately the same way. This is a radish, this could be an oak tree, it could be a bamboo tree, it could be poison ivy. The attributes I'm going to tell you about now are something that every terrestrial land plant shares. So any given plant is separatable into above ground tissues and below ground tissues. The above ground tissues are adapted toward the acquisition of resources carbon dioxide and light, right? So energy and carbon. One mistake that I find in students an awful lot is they think that plants might be able to access carbon and draw it up from the soil. And that never happens. Plants bring in carbon through their leaves, and that'll be important to understand the rest of this talk. The below ground tissues are adapted towards completely different purpose. They're adapted towards bringing water into the plant, and then within that water, they bring all this solubilized material that is also important. Nitrogen, which is needed to make every protein, phosphorus, which is needed to make every molecule of ATP, which is the energy currency, magnesium, iron, calcium, molybdenum, all needed for the enzymes that catalyze the biochemical reactions that make life go, right? People are sometimes interested to find that in order to build a gram of dry material, a plant needs to cycle between 500 and 1 milliliters and 1 liter of water, right? So plants require a tremendous amount of water, which is why irrigation is a big deal, basically because these things are in such low concentrations. So in order to access all of the nitrogen, particularly that one needs, an awful lot of water must be cycled. And that becomes important because the hole in the leaf where the carbon dioxide comes in is the same hole where the water goes out. So they are called stomata. You can see them in this picture under the microscope. This right here is the hole where the water would go in. Around that hole are what we call 2 guard cells. And what the plant does is it very efficiently dehydrates those guard cells, which creates a hole. And then it can also hydrate those guard cells, which closes the hole, right? So the plant has control about the relative openness versus closedness of each one of those stomates, each one of those holes, on an individual level, right? So plants do decision making. It's not the same decision making that we do, but there is intentionality on the action of each one of those cell configurations, right? There's also a great deal of control over the placement of stomata and the density of stomata. So as each leaf is being engineered, a different number of stomata might be drilled into it based on the changing requirements of the plant as it grows. So plants are really interesting, and they're a lot more, I think, dynamic and animated than students often give them credit for. So now let's talk a little bit more about stable isotopes. When I showed you the slide before, I said, don't worry about this, just think of it as a number line. Now let's start to think about what isotopes are doing. So I'm going to talk about carbon-13 and carbon-12. I know many of you already know this, but I want to just spell it out for people that might not be familiar with it. So most carbon in the universe is carbon-12. It's the carbon that you learned about in high school. 6 protons, 6 electrons, 6 neutrons, and it adds up to a mass of 12. About 1% of the carbon in the universe, due to processes within stellar masses, is carbon-13. It's got an extra neutron. You all remember from high school that a neutron has no charge, right? So having an extra neutron in your nucleus doesn't change the way you act chemically. Doesn't change who you bond with or how you react with other substances. It just gives you extra mass. This becomes important for geochemists because we can expect these stable isotopes. It won't degrade or decay the way others do. We can expect it to move through chemical systems identical to the carbon-12, bond and react the same way, but we can expect physical processes that will separate it on the basis of mass to partition any given pool in accordance with the laws that govern that process. So all that solid line box is the ratio in a given substrate. We apply that process to it, and you can look in the dashed box. We come up with a very different ratio in the product. And the process that I've put in there selects for that heavy isotope, and so the value on the number line is going to change. Some of these processes are physical, so diffusion, solubilization, they all impart fractionations, and some are biological. The affinity of a given enzyme can impart a very large fractionation, and we'll talk about both of those kinds because they happen in plants. So here is a cross-section. I drew this a long, long time ago. It used to be an overhead, and it was a really cool state-of-the-art overhead because it had color. Right? And then we took a picture of it, and it became a slide. And we used to go to these conferences and walk around with these slide carousels, if anybody remembers that. And then somehow we scanned it into a computer image, and now it's a PowerPoint. Who knows what I'll be showing it at? You all have it on your Google Glass or something 10 years from now. I don't know. But this is a stomate from the side, so from a cross-section. And basically all photosynthesis is that carbon outside the plant becomes carbon inside the plant, becomes the carbon of the plant, right? So we've got to diffuse through the stomata, solubilize into the cell, and then it gets fixed and turned into sugar. That oxygen gets ripped off, and it's turned into sugar. By the way, when that oxygen gets ripped off, it becomes a waste product. It actually interferes with photosynthesis. It's actually garbage to the plant. And so it's shunted out before it can interact with the biochemistry. And lo and behold, we evolved as little more than a parasite that can breathe that waste product, right? I always love to take the opportunity to tell that to students because they're very invested in this romantic idea that the plants give us oxygen and we give them CO2, et cetera. But the plants are just fine without respiring, oxygen-breathing organisms. They were here before they showed up, and they'll be here long after they're gone. Anyway, so there, of course, have been both theoretical and mechanical descriptions of how the isotopes work. And I'm not going to belabor this, but it's very much dogma within the literature. Most of these relationships were put together in the early 80s. And what they basically say is that the composition of the plant is equal to the composition of the atmosphere, modified by a bunch of physical factors, and further modified by some ecological factors. So the physical factors include things like solubilization into the cell water and also diffusion through the stomate itself. The ecological factors are a little trickier to explain. That stomatal regulation is tremendously important because it's really the only mechanism the plant has to modify its environment at very short time scales. Stomata can shut down. If a plant decides to shut down all the stomata in the leaf, it can happen in a matter of tens of seconds. Right? It's a very responsive system. By opening and closing that door, the plant is trying to accomplish all kinds of things, and none of them really have anything to do with carbon, is the interesting thing. I will show you a little bit more about that. So as the plant, I don't know why this turned into an exclamation point, but I kind of like how jazzy that is. That should be a delta. And I don't know what that should be. When Macworld turns into PC World, like things get jazzier, right? Okay, so all kinds of literature was out there, and even more exists now, talking about how different environmental processes can affect the resultant carbon isotope value of the plant. There's empirical evidence that, of course, if you change the raw material, you get different plant tissue. If you increase PCO2, this was an embryonic idea that people more or less laughed at until we did a whole bunch of work on it. You can get some change in carbon-13. You increase the precipitation at a site. You increase the amount of light at a site. You increase the temperature. This can all come into a change in the carbon isotope composition of the plant. I like to warn people against what comes up as a fairly simplistic brand of thinking that's quite popular right now. People will go to the literature and they'll see, oh, light changed and carbon isotopes changed in the plant. Then they'll run over to the fossil record, measure carbon isotopes, see a change, and say, oh, that means that the light availability in the world during that time must have changed. Right? You see that an awful lot with available precipitation. Oh, carbon isotopes changed. It must mean that the world got drier, or the world got wetter, or the world got hotter, or the world got colder. What's really going on here? It all goes back to the stomata. So if a plant wants to move, if it wants to take its leaf from here to here, because it has a guess that over here is sunnier than over here, it does that by dehydrating and rehydrating and dehydrating and rehydrating. It doesn't have muscles like we do, but it opens and closes those stomata. and dehydrates and rehydrates and slowly moves the tissue along. And you've probably seen that if you've ever seen morning glories or you've ever seen sunflowers track the sun. That's exactly how it happens. There's these very sensitive cells right here that act to move the plant. And as the plant does that with the stomata, that's how we get an effect on the carbon and then a change in the carbon isotope value, right? Water is the same way. So as precipitation goes down, the plant opens the stomata in order to pull more water in. Plants are basically capillaries evaporating from the top, and we get a change in carbon, which feeds back into the change in carbon isotopes, right? So this all goes back to stomatal mechanics. So what we wanted to do was look at the phenomenon of this carbon fixation upstream of all of this after-the-fact stomatal regulation. And here it is just presented a different way. So the difference between the plant and the atmosphere, I'm going to show you a lot of big delta because different plants have different compositions. Atmospheric composition can change. So I'm going to talk about the net fractionation between the two entities, modified by those physical factors, further modified by that ratio of openness to closedness. And I just want to say that this is really very hard established dogma when we took it on. This is still, and this is as of recent most, Google search or whatever I did, Google Scholar. These papers were written in the 80s and they're still in the thousands of citations. People are really, really cemented in their interpretation of the phenomena according to that paradigm. Today, changes in TOM, so this is changes in terrestrial organic matter from the fossil record, is commonly interpreted as either a change in the substrate, which was an idea that we pioneered and somewhat no longer believe, or we've qualified it to a certain extent. I always tell students, if you live long enough, if you stay in the business long enough to prove yourself wrong, you've done a good thing, right? That's really the definition of success in some ways. Changes in water availability, again, because of that precipitation effect, or changes in vegetation types, which goes back to the C3, C4 thing, which I will talk about a little bit later. C4 versus C3, this two end member mixing, which I'm actually going to talk about some problems with that. That's relegated to relatively recent strata. So the C4 is an extra enzyme, and we're going to talk about how that works. But looking for enzymes in the fossil record isn't particularly doable, right? So what people look for is the anatomical specificity associated with the utilization of that enzyme. The earliest that we think we see that anatomy is around 12 to 15 million years ago. So the C3, C4 question is really relegated to the Neogene, the rather recent Neogene. If you ever see a paper that says C4 has popped up in the distant geologic past, you got to take that with a huge grain of salt. Okay, so when we first looked at this, back to the original shift that we saw in the Columbia rocks, we were thinking of what in the heck does this mean? We went to the literature, and we looked up every agricultural study that we could find, everything that we could find that measured the composition of the plant, the composition of the atmosphere, and the composition of the CO2. Right? And this was mostly in the agricultural literature because people had been using plant tissues for ag applications for a long time. This was all the literature that existed in the year 2000. So that was before the widespread use of the EA. So you can imagine, it's so much easier to make data now than it used to be with carbon isotopes particularly. What we saw was a correlation between, lo and behold, what was in the atmosphere and what was in the plant. We saw it had a slope of 1, and we could regress it with relatively high accuracy. And we saw basically an offset. So we saw smears within individual communities due to those stomatal regulation things. Everybody was working with its stomata in a different configuration in these farm fields and all these various places where the literature came from. But on average, it broke down to this standard offset between the atmosphere and the plant. So we thought, hey, if you change the composition of the plant in the fossil record, and you're looking at a substrate that you know involves the accumulation of hundreds or thousands of individuals, right? Then we can fairly accurately probably reconstruct the composition of the atmosphere from the composition of the plant. So we interpreted that big spike as a real kick to the atmosphere during that time period. We didn't think CO2 played a role, and that was because of this graph. So if we looked at the PCO2 value that each of those experiments were done at, and the vast majority of them were done under modern conditions, right? During those days, there were actually just a very few studies that looked at elevated CO2 and also did carbon isotopes. But based on what we could see at the time, we thought that CO2 was not playing a governing role. We thought that there was no reason to believe that CO2 would really make a big difference, interestingly. And so we set up some experiments. What we wanted to do was to really nail down this relationship and quantify it, because we wanted to correct for atmosphere using plant. We wanted to know very specifically what that offset should be. So we cooked up some experiments where we were going to change the composition of the atmosphere and grow plants. Right? We're going to do this in a controlled situation, not from the literature like this stuff, but we were going to grow the plants. The easiest way to change the composition of the atmosphere was to add a bunch of CO2 of this really light description, right? So we would just add CO2 at these increments, which would then change the atmosphere toward the composition of what we were adding. And we were going to measure the plants, nail down this number. It was all going to be great. It didn't work out that way, but we got to something better. To our credit, we did have some appreciation of just how important CO2 was to a plant. So CO2, you remember my graph where it comes in the leaves, other things come in the roots. CO2 is a little like money to plants, right? So CO2 comes in, it gets fixed as carbon, as a real rudimentary sugar, and then the plant can do any number of things with it. It can use it to build a protein, it can build a bigger sugar, it can make an enzyme, it can even burn it for glycolysis if it's really desperate for energy, or it can leak out the side due to oxygen contamination, right? So once carbon is gained, all kinds of different things can happen. If money dropped down from the sky and I raised the salary of each one of you, or let's just say your wallet, if all of a sudden I snapped my fingers and your wallet or your purse had double, triple the amount of money in it that it does right now, that would probably change your activities for the rest of the week, right? And probably every darn one of you would do something different with that extra money, right? Some of you would buy a lottery ticket. Well, you'd have to cross state lines to do that, right? Am I right? Some of you would risk something and gamble something. Some of you would go take a class in windsurfing. You'd try something new. Some of you would pay off your bills. Some of you would eat an extra dessert that night, etc., right? And the plant world is a little bit like that too. So what we've seen during geologic time, and again, this is based on proxies and models, and I'm actually going to come back to this because we think we've got a really good window on CO2. But the thinking is that CO2 has changed quite a bit over geologic time. So this is the time of the dinosaurs, which we regard as a high CO2 era. And the time of the Pennsylvanian and the Carboniferous, when we had vast coal fields, we also see that as a time of high CO2. This is the Permian-Triassic, the great, great extinction that sort of obliterated most biogeochemical cycles, except for a lot of plants. So that has changed a lot through time, and it matters, right? So again, if your budget changes radically, that could very much influence the distribution of traits that we would find in this room. This is the volume of basalt from a few different places that we see during time. So this is part of the Mesozoic up until about 50 million years ago. And what you see here is the volume of flood basalts. So at various times during the Phanerozoic, you have these massive volcanic eruptions. So a lot of India is actually one big lava flow. So there have been periods where there's been massive volcanism, which of course drives up the CO2 content of the atmosphere. And during the Phanerozoic, there are some really big notable flows. If we look at the evolution of plants, we see that some very critical adjustments within the major lineages happen not totally distinct from where these critical periods are. So it's not unreasonable to say that CO2 would have a fundamental effect on how plants operate. It's also not unreasonable to say that plants must have a way of dealing with very high CO2 since they've been so successful and persistent for so long that they have been exposed to these fluctuating values. So when we set out to do this, we were also the first people to go up to very high levels in growth experiments. Growing plants at high CO2 is not a new thing with me. And we talked a lot last night about the ramifications for agriculture that we've also worked on at these high levels. But today we're talking about isotopes. Most of the work that governs what plants get grown comes out of the Intergovernmental Panel on Climate Change studies that came out in the early 90s that tried to predict how CO2 levels might rise over the next 50 years, over the next 100 years, et cetera. And the value that they came up with, the sort of optimistic value they came up with, was 550 for the year 2100. At that point in time, a huge flurry of ecological studies were started in order to investigate the effects of 550 on plant growth. There are implications, of course, for forestry, implications for agriculture, also implications for medicine. So those are the three main ways in which we use plants today. The basis of a lot of our medicines, wood, and also food. If we look at that in terms of geologic time, we see that today we're looking at about 400. This was current in 2009. Prior to the Industrial Revolution, things were about 300. There were a few growth experiments that went up to 750, but in the Cretaceous, so during the time of the dinosaurs, or throughout the Mesozoic, we believe, based on the best geological evidence, that things could have been as high as 15 times today. So what we wanted to do was work at those very high levels that plants have possibly seen in the geologic past, particularly during the Cretaceous, which was such a critical time for plant evolution. My colleague that was working in Colombia was very excited to be working there and worked there at great personal risk because she was interested in the first flowers. So you walk around and almost everything you see today is a flower, has a flower in it. Even grasses have these little tiny flowers that you need a magnifying glass to see. Everything outside the broad conifer forests is actually a flowering plant. Flowering plants only evolved about 125 million years ago, we think. So 125 million years ago, this radical new means of reproduction came in, took hold, and that's 95% of everything you see nowadays. So she was very interested in the Cretaceous, also a time of very high CO2, and I was interested in the isotopes. So how were we going to do this? We were going to grow plants at those very high levels of CO2. We thought the CO2 would make a difference, but we were sure we could also measure the carbon isotope composition of the atmosphere provided to the plants, the composition of the plants themselves, and we were going to compare those two things, right? We did this by building chambers using things from Home Depot. We started with things as small as a shoebox and things as large as part of a farm field. We engineered these ourselves through lots of trial and error. It took us years to get chambers to the point where we could control CO2 levels within a few ppm. We didn't have a lot of robotics when we started out. I often had students with a sleeping bag sleeping here so that they could monitor conditions on every 10 minutes, since those stomata are adapting in real time, right? So if you want to solve a really good problem, you're going to have to do it without machines, at least at first. Started doing this with a teaching grant, actually, because I wanted to get a lot of students involved in growing these things. This was one of the first chambers. It was upright like a bookshelf. Now they look like coffee tables and stuff. So what could we control? We could control the growth stage by picking the right plant. We could control how much cations were supplied. We could control very closely how much water was supplied and what the relative humidity was. We could control how much nitrogen and phosphorus and nutrients were delivered. And of course, we had very good control over light, intensity, and duration. We controlled very very closely all these things that had been shown in the literature to have stomatal reactions. And then, of course, we varied the CO2. I want to show you a couple movies of plants that we also take time-lapse photography when we grow a lot of these plants, mostly because it makes for an interesting talk. People love to see plants grow, and it also helps me bring home my message that plants are far more animated than we give credit for, that we tend to somehow think of them as very passive organisms when they're not at all. As you can imagine, in these chambers, there's no wind, right? It's completely closed. Not completely closed, it's actually at positive pressure, but there's no wind moving through there to move these guys. So I'm going to show you a few days worth of growth, and I think you'll be surprised at how much they move and also how much they interact with each other, particularly those two in the middle. So again, there's no wind in there. What they're simply doing is hydrating and rehydrating themselves and experimenting with capturing the best light for their growth needs moment to moment. You'll see this guy kind of laying down and spreading out, and then you'll see this guy kind of lean over and shade him, and then you'll see divestment in this more shaded leaf. He's sort of not bothering to push that one up toward the light anymore. And then this guy over here doing his own thing is minding his own business and growing much bigger than the others. We watch these movies a lot. So I'm going to tell you about experiments where we've grown a total of 7 species, hundreds and hundreds of plants, 15 different levels of PCO2, and we measure the isotopes on all of these things. And we also do some nice photography. I'm going to tell you particularly about a couple species. Of course, we pick our species very carefully. We can't grow every single plant out there, and so we're very careful to pick model species that we think the processes are generalizable across great distances within the plant world. We pick Raphanus because there's a lot of work that's been done on partitioning and development, which I talked about yesterday more. We grow Arabidopsis because, of course, there's a known genome, and there's a huge amount of CO2 literature at those lower levels. And here's what we saw. Here's the interesting thing about what we saw. You remember that graph early on that said no relationship between CO2 and carbon isotope value? Doing these experiments carefully showed us that there's a very well-defined relationship between the net carbon isotope displacement and PCO2. And that we saw it in N-alkanes, we saw it in specific compounds, we saw it in above-ground growth, below-ground growth, and we saw it across species. And these are large effects. So this is 22 up to 28 6 per mil effects, sometimes 9 per mil effects, very, very strong effects, only the CO2. Remember the great pains that we took to control environmental conditions so that we can't attribute any of this to the stomata? This is all a CO2 effect, and I'll tell you why we think that is. The first thing we looked at when we saw this, and it took us a while to publish this. It came out in GCA, in Geochemical, a little while ago. But the first thing we saw when we published this was, oh my gosh, we could use plant isotope composition to solve for PCO2. So figuring out changing PCO2 is a bit of a holy grail in geochemistry. We have a lot of methods to solve for PCO2, but none of them are very specific. What we saw here was huge isotopic sensitivity over very small changes relative to geocarbic, et cetera. And I'm going to show you some very good news that we think that this particular technique is really going to take hold very soon. But how to do that, right? So each one of these has a different sort of y-intercept, right? So we have different offsets according to different species and compounds. What we did was we looked at the slope of any given 2 points here as a continuous function. I'm going to show you that. So what I'm going to show you is graphed the changing slope of this function compared to all of them throughout each of those experiments. The other nice thing about that is we could compare it to things that have been done before. A little bit about why this is happening. So C3 photosynthesis works like this. And again, I'm talking about C3, which is this type that's been around for a great deal of time. C3 photosynthesis turns CO2 into this common sugar. What we think is going on is that this sugar leaks out during times of high CO2, and that's called photorespiration. But we believe that the elevated CO2 serves to plug this leak, and I'll show you why. Here's our data compared to a poor little Arabidopsis mutant that's not able to perform photorespiration. It's deficient in the glycine decarboxylase. And you see that as we go up to this very high PCO2, we're actually going toward that point that it's starting to look like that mutant that doesn't bother to do it. The other thing is glycine and serine are some important metabolic intermediates of the photorespiration pathway. And we can see that at times of high CO2, you see that lower fractionation effect. We're also seeing very high glycine and serine. So that's additional independent evidence that the photorespiration cycle is turning off. Let me show you now a generalized relationship based on slope that'll take us from any plant material to the PCO2 value. So these aren't error bars. These are calculation from the one level to the other level and the slope in between. And so this is a very, very tightly strung relationship quantitatively. It works very well. When you're a woman in this business and you discover something very new, the first thing people ask is, why didn't anybody see this before? And if you really want to piss them off, you say, they did, they just didn't know what they were seeing. So the black data is a lot of studies that were done mostly at lower PCO2, where they did 2-point studies, so they had a slope, right? They went from low PCO2 to high PCO2, and there was a slope associated with that fractionation. Lo and behold, if you plot all the previous data, you get the same relationship to a very high accuracy that we were measuring within our chambers. This falls apart a little bit at low PCO2 in the literature. So our stuff only goes up high. And so one thing that we've got resources for, we've got a grant for, is to work at these lower values. So stay tuned for the results on that, because that's actually a very sensitive part of the curve. This could work very well at subambient. How to prove this works? So we're kind of doing a CO2 experiment today. CO2 is going up all the time. So if our method works, we should be able to see this in real time. This is a tree ring. These are our measurements from a Manoa tree in Hawaii. This carbon isotope through time. And it's about 80-some years of data. And what you see is, since about 1980, you see this decrease in value. And if you were a water person, and many people are out there, you would interpret this as a wetting phase. Oh, carbon isotopes are getting lighter. There must be generalized wetting in the environment. The problem with that is there isn't, right? So we also know precipitation records for that period, and we know that things have more or less stayed the same. If we then correct this for what we know the PCO2 effect is, according to how PCO2 has changed, using that S-curve relationship, we actually, based on the increase here, we actually get something flat that actually lines up pretty well with the precipitation record. So again, we're not saying precipitation doesn't matter. We're saying that by correcting for CO2, we can actually get a much more finely tuned quantitative relationship with water availability, and that's pretty neat. The other way in which this matters is that we believe it explains this discrepancy between the marine record and the terrestrial record. And this is the PETM. We recently published this in Nature. This is the PETM shift. So like the Columbia shift that I told you about, the terrestrial goes much further down than the marine. And that, we believe, is because the marine contains just the shift in substrate, while the terrestrial contains shift in substrate, but also that PCO2 effect. This leads to an interesting scenario where we can use the PCO2 effect to calculate, I call this delta, delta, delta carbon-13. It's my stable isotope sorority, right? So the change in the total offset will actually, if we have a starting point, allow us to calculate the change in PCO2. So that was an interesting paper for the PETM. I'm not going to belabor it, but it allows us to look at specific CO2 rise scenarios during that time period. Here's a last interesting bit of evidence that I'm going to show you. Another CO2 experiment that the world has produced is the change in CO2 across the glacial interglacial cycles. So this is the last glacial maximum. And we have looked at plant fossils from around the world, 24 sites globally. And what you see if you look at delta, 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 again, that's that change in the net fractionation. You see that there's been this rise across the last glacial maximum. We also have CO2 data for that period, right? The ice cores exist. We get very fine scale ice core data for that period. So if what I'm saying is that you can use that S-curve to calculate CO2, that should match up with the ice core, right? If it works, let's see how well it does. And you can badger me about my smoothing functions and stuff like that after we're done. So here's the ice core record. And here's just the last little bit we're going to talk about. This is what it looks like going through. This is what it looks like. The green data is what it looks like if you do this with leaf fossils. And the brown data is what it looks like if you do this with terrestrial organic matter. Right? So we're doing pretty well in terms of being able to trace the global history of CO2 according to the ice records. I'm going to tell you a little bit about C4, and then we're going to wrap up. So this is C3, carbon dioxide becomes sugar. C4 is basically just a front-end bit of biochemistry that goes on to C3. C4 takes carbon dioxide, turns it into sugar, and then dissociates back into carbon dioxide and feeds it into the C3 system. That's all C4 does. It's this extra piece of photosynthetic machinery that precedes general C3. It's expensive. That's why not all plants do it. You need an extra enzyme, you need a bunch of intermediate energetics, and you need ATP to repair it. Let me show you a C4 plant that we grew, just because I really like this guy. He grows for a long time, and then he kind of explodes in terms of his biomass. Watch right here, and you'll see that layer fill out really quickly. Whoa. Okay. That was about a week's worth of growth. What do C4 plants do? Because again, the biochemistry is different. That leaky pocket should work differently in these guys. So here's C3 plants. Remember that hyperbolic function, the way it looks. Here's some C4s. Those are grasses. This was the accepted, assumed pattern for most C4s. But what we saw in our broadleaf C4s is that a linear response, we don't know when that levels off, actually, which is really interesting. We've done a few more C4 species, and the broadleafs seem to also follow this. What's important for this is that often you'll see in the fossil record, people will measure carbon isotopes, and they'll say, oh, this must result from a contribution of C3 and C4 plants. And if that value changes, they'll say the relative proportion of C3 and C4 is changing. That's based on an end-member mixing model. The danger with that is if they're also working across a period of changing PCO2, the end members may be changing as they do that experiment, right? So this casts an awful lot of complexity into the standard model of end member mixing that's in very common use for C3, C4. Future work, if you ever want to have me back and hear something about our future work, we're going to do this with ferns. We're growing them from spores, which is a really sucky piece of hard work. Most of the time, if you grow ferns, you take a snippet and you transplant it. Trying to get these guys to go reliably from a spore is a whole new set of growth experiments. It takes months and months. So once we find the right martyr student for that particular project, we'll have some great results. But of course, ferns go way, way far back in the fossil record. So if this particular method works with ferns, we could get some very good Paleozoic PCO2 estimates, and that's going to be worth its weight in gold. We know very little about the absolute value of PCO2 for very early times. One last slide, and then I will be glad to take questions. One thing I like to leave people with is a reminder that the Earth we know today is just a snapshot of what the Earth can be. So life has been on Earth for around 4 billion years. For the first several billion years, all the ecosystems on Earth were limited to one-celled organism. That's part of the reason why we're looking for microbes on Mars. If you happen to visit Earth over the last 5 billion years and you see life, close to 3 billion years of that trajectory would have been microorganism ecosystems, right? And then, since multicellular life evolved, we've had several different Earths. So in animals, we've had food webs that were based on trilobites, which is an organism we don't have anymore. We had 30-ton reptiles, which we don't have anymore. We've got 5-ton mammals, during the recent past. The Earth system as we know it is really relegated to the last few thousand years. And in the plant world, it's similar but different, right? So seeds only evolved a little while ago. So can you imagine forests with no seeded plants in it? Can you imagine a world without flowers? Almost everything around us today is a flowering plant. And then there are great examples like the dragonflies with 1-meter wingspans from the Paleozoic. So what I like to tell people about is that geology, reconstructing the past, is an awesome way to stretch your imagination, to think about what the world can be. But I also like to caution people, this is probably the most important thing I'll say all day, is that ancient organisms did not exist in order to provide faithful paleoclimate estimates for future paleoclimatologists. They were living organisms with an active biology trying to solve 5 problems: growth, maintenance, storage, reproduction, and defense. We have to start looking at things from their agenda. What were they trying to do? How did they react with the environment? How does that give us a window on the past? It might not be what we think we want to know, but it might be something a lot more important. Thank you so much for your attention. I know I went a little bit over, and I would be delighted to take your questions. Yes?
Here's again, you're talking at that plot of your early data.
Yep.
We have that negative carbon excursion.
Yep.
And what is your...
What happened in the Aptian?
With that now based on research?
I've decided I don't care. No, it's funny because a lot of people know me as an Aptian expert, which is really funny because for years, like I would go to AGU, and I remember there was one year the guy was standing on a folding chair yelling at me for showing this data. How can you do this? Don't you know that plant fossils don't mean anything? You can't do this. And now I'm kind of like the Aptian lady, and it's really easy for me to publish on this. I sort of get to be right? So what does this mean? At the time, we had this theory, but I've kind of stopped working on the Cretaceous because I said to myself, I don't want to go another 20 years arguing with the same people about the same thing, right? So I started working on food. I started working on a bunch of stuff. Here's what I think about the Aptian. This is also interesting because there's a big anoxic chapter in Earth's history there. There are all these reduced layers in the ocean sediments. And it looks like the ocean was all devoid of oxygen for a fairly long time period, kind of all at once. It's very, very dramatic if you look at the marine sediments. What we thought, when we thought it was just the atmosphere taking this big punch, we figured that you needed a methane hydrate release to impact things so much so quickly, simply because this is like 7, 8 per mil, right? And we thought, well, that was reasonable because you'd have the methane come up, swing things isotopically, it would oxidize to CO2 and suck down all the oxygen. Right? This got muddy because of the stratigraphy. I'm basically assuming these are all the same event. This isn't like the PETM or the Paleocene or the Pleistocene where you can actually line these things up. This is the Cretaceous. So each one of these things is plus or minus 1 million years, right? So we have to assume this is all the same event in order to make that idea. What we came to is that the difference between this and this might be due to PCO2 change. So now we have a scenario where we have to reconcile not just that change in isotopes, but also a change in PCO2. So we have been able to further constrain it and say that whatever did this isotope slide must also have yielded a PCO2 change of this magnitude. Right now we're trying to see if that's more in line with the methane hydrate or with the volcanism sort of line of contention. I think that'll probably devolve into yet another argument over timing. But our calculations show, again, that methane hydrates are probably the the most acceptable answer. With the anoxia, I don't think there's any way to think that it's not methane hydrates. I don't know how many more ways and times I can show it, but that's sort of where I am with that particular problem. Where's all the lava, right? So yeah, I mean, 2/3 of the Earth would have to be lava or something we tried to calculate, but you get in trouble by making those calculations, right? So that's sort of where we are with that particular problem. That's a long answer to a short question. Anything else? Thank you so much for your attention. I'll be standing up here if there's anything you want to talk about.